Maria Elisa Sanchez, pronouns she, her, was born and raised in the Ecuadorian Andes and has specialized her research in studying how human pressures affect mountain peatlands. She studied environmental engineering at Universidad San Francisco de Quito, followed by a master's in applied ecology from Michigan Technological University, and is currently studying a PhD at the University of Saskatchewan in Canada. And she's based in Canmore, the traditional territory of the Blackfoot Confederacy, Tsuut'ina and Stoney Nakoda First Nations. And she is studying how peatlands and the Canadian Rocky Mountains are affected by a change in winter precipitation due to climate change. She serves as the Associate Coordinator of Youth Engaged in Wetlands, committing to help with the protection, conservation, and wise use of wetlands. She is also an active volunteer and instructor with Inspiring Girls Expeditions, seeking to empower girls to lead and succeed through science, outdoor, and art education. And if you want to follow Maria on Twitter, which I would highly recommend, you can follow her at ME_SanchezG. Maybe we can put that into the chat as well. But yeah, a great way to stay connected with Maria and her fascinating work. And so I will turn the time over to you, Maria.
Thank you, Becca, for the presentation and for the introduction, rather. And I'm happy to be here and to be presenting this work. So good morning, afternoon, or evening to all of you, depending on where you are. I hope I can make your time listening to this seminar worth it. And I will tell you a story about an experiment we carried out as part of my PhD research. It was a reciprocal transplant experiment that revealed some interesting insights in carbon sequestration in mountain peatlands. And I see that there are a lot of concepts in this title, but rest assured, we will unpack that, what all that means as we go through the presentation. So first of all, before I talk about my work, I want to acknowledge that I'm speaking to you from the land, traditional land of the Washachu Ite, and my research was carried in the traditional territories of the Blackfoot Confederacy, the Tsuut'ina and Yahe Nakoda First Nations, and Métis Region 3. I am grateful for this land, for the mountains and the water, and all the living beings that allow these observations to happen. And I'm also grateful for the presence of these communities who have observed, learned, and cared for this land since before these peatlands even started to accumulate peat. Since the start of the industrial era, human activities have released large amounts of both methane and CO2 into the atmosphere. These two greenhouse gases have positive radiative forcing properties, which makes them a key component of climate change. Anthropogenic emissions are predicted to increase in the future, and in phase of increasing carbon emissions, there is an important need to quantify carbon from any potential source, as well as the generation of knowledge around carbon sinks in nature. As part of the multiple ecosystem services that wetlands provide, they play a critical role in global carbon dynamics. Enter peatlands. Peatlands are wetlands that accumulate thick layers of undegraded plant material under waterlogged and anoxic conditions. In most peatlands, the net ecosystem exchange, which is the net carbon uptake by the system, outweighs the carbon losses to the atmosphere, and hence their carbon storage capacity. Peatlands are so efficient in storing carbon that they cover only 3% of Earth's land surface, but represent approximately 30% of the global soil carbon pool. And what allows them to be so efficient compared to a forest, for example, is this delicate balance between soil, vegetation, hydrology, and temperature. However, changes to this balance can cause shifts on the ecosystem's carbon dynamics. Peatlands exist everywhere around the world. They can be found in the tropical rainforests and in the Arctic, but are most extensive and prevalent in the temporal boreal zones. Peatlands found in the mountains may not be as vast as in most flat regions, but they exist in most mountain ranges in the world. The peat profile in mountain peatlands is marked by sediment layers. Given their location in confined valleys, sediment is transported by snowmelt water, wind, and landmass movements or volcanic events. Like this peat core that was extracted from a peatland in the Ecuadorian Andes that has a thick layer of ash from a volcanic event in the past. These little happy pockets are an important part of the ecosystem as they bring heterogeneity to the landscape, harboring different species than their surroundings. The Canadian mountains alone harbor an estimate of 13,000 square kilometers of peatlands. Peat in the eastern side of the Canadian Rocky Mountains started to accumulate after the last glaciation, and the ages range from 4,000 to 13,500 years before present. Global perspectives of climate change predict changes in temperature and snow water equivalent in mountain ecosystems. In the Canadian Rockies, mountains at higher elevation may experience increased winter precipitation with thicker snowpack, whereas lower elevation mountain ecosystems may see less snow cover extent and depth. This is particularly important for mountain peatlands as mountains are being disproportionately affected by climate change. So what does our reduced or increased snowpack mean for peatlands in the mountains? Well, there is a lot of research regarding the interactions of snowpack and soil temperature in forested soils, borough peatlands, and permafrost. However, not a lot of these studies have addressed mountain peatlands specifically. Another part of my PhD research is aiming to look at these interactions more closely. What we do know is that seasonal ground ice is regulated by air temperatures, snow depth, and duration of cover. Snow has insulating capacity. So a thick layer of snow allows the soil to stay at a constant temperature during the winter, preventing it from freezing. Without that insulating layer, soil or peat is more prone to frost. So in a way, the quantity of snow and the timing of snow melt during winter controls carbon fluxes by controlling the seasonal ground ice and both the hydrological regime and the integrity of the vegetation and microbial communities of the soil. This is the background of where our research comes to life. Our hypothesis is that the carbon fluxes during the growing season are influenced by the conditions of the preceding winter. So the objective of this study was to determine whether peatland gas-based carbon sequestration during the growing season is advantaged by soil remaining thawed in winter. And we divided into three smaller objectives. The first one is to quantify the differences in winter conditions between sites. The second one is to determine the differences in carbon fluxes between home and abroad transplants from each site. And the third one is to estimate carbon sequestration throughout the growing season for transplants in both sites. So on to the research design and methods. We worked on the eastern slope of the Canadian Rocky Mountains with two peatlands, one in the foothills called Sibbald, and the second one above tree line called Helen. Both sites are equipped with a meteorological tower. Sibbald Fen is located in the foothills of the Rockies in Kananaskis country with an area of 1.4 square kilometers and an average elevation of 1,480 meters and a peat basal age of 13,500. Sibbald's meteorological station is located in the southern corner of the peatland, as you can see here. Helen is a fen located in the Helen Lake watershed in Banff National Park. This peatland is much tinier than the foothills with an area of 0.01 square kilometers and a mean elevation of 2,365 meters and a basal age of 3,820. Helen Creek's meteorological station is located in the upland, approximately 2 kilometers away from the peatland. Several approaches are taken when manipulating the soil temperature in snow-covered environments, from in situ snow exclusions to soil core freezing and thawing in controlled laboratory environments. We chose a reciprocal soil transplant between two sites at different elevations as a way to allow the natural air temperature and snow depth differences to expose the transplant to contrasting climates without tending the transplant over the winter. Providing two peat soils with different year-long conditions allow us to simulate possible changes in the future. So about the peat transplants. We transplanted the peat in September 2019, and a big shout out to Becca who volunteered with us to make this happen. We chose two sections of both peatlands that have similar vegetation type and minimal microtopography. Minimizing these differences leaves climate as a primary variable differing between the two peatlands. The transplants were randomly located within similar areas dominated by carex, salix, and moss species. The transplants are cylindrical blocks of peat and surface vegetation of 30 by 30 centimeters. In total, we transplanted 5 blocks of peat from Sibbald to Helen and vice versa, which are called abroad. And we also transplanted an additional 5 peat blocks into their home peatland to control for any inherent variation due to the transplanting, which is called home, or home over here. We randomized the location of the transplants in the dominant shore vegetation of each peatland with at least 50 centimeters of distance from each other. We then allowed the transplants to overwinter in their new locations. The naming convention for each treatment type in our reciprocal transplant experiment follows as such. The first letter of the site of origin, so this one is Helen, by the first letter, followed by the first letter of the site where transplants overwintered. So this one came from Helen and overwintered at Sibbald. The ones that stayed home came from Sibbald and stayed at Sibbald. We allowed the transplants to overwinter in the new locations while we measured environmental variables such as soil and air temperature, snow depth, radiation, among others, and visited them the next summer for carbon flux measurements. Once we were able to visit the transplants in the growing season, we measured CO2 and methane fluxes during the day using the static chamber technique over each of the transplants. We measured CO2 fluxes with a cylindrical clear acrylic chamber equipped with a fan connected to an infrared gas analyzer in a closed path with deployment time of 124 seconds. We measured the CO2 values for net ecosystem exchange, or NE, with the clear chamber. NEE includes both the ecosystem respiration, which is what the plants and the microbial community is respiring, and the gross primary production, which is what the plants are taking up. We then covered the chamber with blackout cloth to measure the ecosystem respiration. We also carried out these measurements at 2 different levels of photosynthetic active radiation using shading. We determined the gross primary production as the difference between NE and ER. We also measured methane fluxes using cylindrical PVC chambers equipped with fans with a deployment time of 45 minutes. We collected 20 milliliters of gas from inside the chamber into vials, and we took gas samples every at times 0, 15, 30, and 45 minutes, as well as ambient measurements previous to deployment. We send the methane samples to be analyzed at a lab. After some delays and hurdles due to COVID, we were able to go to the field and actually measure carbon fluxes. Though our visits were limited, we measured carbon fluxes for a total of 4 times on each site, from July 20th to August 21st at Sibbald, and from August 29th to September 18th at Helen. On to the results. For objective 1, which is quantify the differences in winter conditions between sites. Here we have a graph of environmental variables for both sites. This data ranges from September 2019 to October 2020. Note the dotted line marking the date of the transplants on each site, and the dashed lines that mark each of the 4 visits to measure carbon fluxes that delineate the study period on each site. What I want to point out from the graph below is the marked difference in snow depth between sites. A maximum depth of 20 centimeters in Sibbald versus almost 2 meters in Helen. I also want to point out the marked water table drop, which is marked in black, the line in black, for Sibbald for the 3rd and 4th date, compared to the sort of constant water table for Helen during the study period. Although it isn't very clear from the orange line here, the soil temperatures at Sibbald drop below 0 for most of the winter, whereas at Helen it doesn't. And this will be more evident in the next slide. So here we have 2 tables of comparison of environmental variables between sites for both winter up here and the study period below. For the winter period, I want to point out that despite the consistently colder air temperatures, consistently cold temperatures at Helen, the soil remains above freezing for most of the winter, likely due to thicker snowpack. And then for the study period, the water table is statistically the same for both sites, regardless of that drop that I pointed out in the middle of the study period. However, air and soil temperatures are different between during the study period between sites. And this is something to keep in mind for later. Just to have a visual of these differences in snow depth, we have here pictures for both sites in winter and in summer. The takeaway from this section is that winter conditions are quite different from both sites, particularly for snow depth and soil temperature. Summer air temperatures are also different between sites. So for objective 2, determine the differences in carbon fluxes between home and abroad transplants for each site. So we're going to start with methane fluxes. Our sign convention for both CO2 and methane fluxes is that the negative fluxes indicate an uptake by the ecosystem, while positive fluxes represent a release to the atmosphere, okay? So in here we have positive fluxes, meaning a release. The analysis we did here was a mixed ANOVA, and I considered the treatment as between subjects factor and the date as within subjects factor. And I want to point out that there is a significant interaction between date and treatment on methane flux, which means that the impact of treatment in the fluxes depends on the date. We followed up with pairwise comparison between treatment levels, but the pairs that are we're more interested in comparing are the home versus abroad for each site, right? So we want to compare the Helen at Helen versus the Helen at Sibbald, or the Sibbald at Sibbald versus the Sibbald at Helen. For example, there's only on date 4 that Sibbald at Sibbald is significantly different than Sibbald at Helen. I also want to point out the drop of methane fluxes on date 3 and 4 for the transplants that were at Sibbald. So remember that drop on the water table that I pointed out before? This reduction in the emissions can be directly related to a drop of the water table, which increases the aeration of the soil and reduces methanogenic activity. For CO2 fluxes, I split them into NE, ER, and GPP. So we did the same mixed ANOVA analysis for all 3. For NE and ER, both have significant interaction between date and treatment on CO2 flux. The pairwise comparisons for NE and ER show us that there are no significant differences on any date between Helen at Helen and Helen at Sibbald. However, the NE of Sibbald at Sibbald and Sibbald at Helen pair are significantly different for day 3 and 4, and on day 3 for ecosystem respiration. For gross primary production, there is no significant interaction between date and treatment, with a significant main treatment effect, which is why I chose to show the results combined across dates. I want to point out the significant difference between home and abroad at Sibbald, with a larger magnitude of the GPP flux on the transplants from Sibbald that overwintered at Helen than those that stayed home. So the takeaway from this analysis is that in most cases, the impact that exposing peat to different winter conditions has on the methane and CO2 fluxes depends on time, likely due to changing conditions. This also allowed us to see that there are differences between the home and abroad transplants, although those differences are not constant through time. This is a great way to understand what is happening in this window of time, but it seems that it would be really helpful to know what is happening during the whole growing season using the information that we gathered. So for objective 3, to estimate carbon sequestration throughout the growing season for transplants in both sites, we did some modeling. So for this section, I'm going to present to you some preliminary data, and this is a work in progress. So we used the data we collected during the study period and the main environmental variables influencing each flux. We modeled the fluxes for the whole growing season on each site. And I want to point out that the growing season at Helen is about a month and a half shorter than that at Sibbald. So we modeled GPP using measured PAR values from the meteorological station. We modeled ER using soil temperature at 5 centimeters of depth using the Lloyd and Taylor respiration model. NEE is the addition of both fluxes. And I'm still working on modeling the methane fluxes, so stay tuned for that. We then use the model values to calculate the cumulative fluxes for each treatment type. This graph shows the NEE, ER, and GPP for treatment type with the zero-axis line in black for reference. I want to point out that the positive cumulative values for NEE in red for all treatment types suggest a loss of CO2 to the atmosphere. This graph puts the NEE together for all treatment types. This shows the daily fluctuations in fluxes in those tiny spikes. So first, we see that the transplants originated from Sibbald accumulated CO2 initially in the growing season for 24 days for Sibbald at Sibbald and 27 days for Sibbald at Helen, whereas the transplants originated from Helen emit CO2 since the start of the growing season. Transplants that stayed at Sibbald, regardless of their origin, emitted more CO2, partly because of the longer growing season. If we compare the slopes for Helen at Helen, so here, and Helen at Sibbald, they look very similar, which leads us to think that the length of the growing season is the primary difference between the cumulative fluxes. However, for transplants that originated Sibbald has a steeper rate of NEE emissions compared to Sibbald at Helen. And that this could mean that the change of environment to a frost-free soil and cooler temperatures during the growing season reduced the loss of CO2 to the atmosphere. So the main takeaway for this section is that the change of environment for the Helen transplants did not have a large effect on their fluxes. However, Sibbald transplants that spent their winter abroad had reduced CO2 emissions over the growing season due to both reduced emission rates and shorter growing season. So to conclude, I'd like to bring the main objective back. So to determine whether peatland gas-based carbon sequestration during the growing season is advantaged by soil remaining thawed in the winter. It seems that overwintering in an environment without frost during winter allowed the transplants to emit less CO2. However, this effects might be masked by the length of the growing season and the differences in air and soil temperatures between sites during the summer. And that past winter conditions impact carbon fluxes during the following growing season, which has implications in the changing climate. So the next steps for my research, as I mentioned before, as part of my research, I will be further studying the relationship of the changing winter conditions in Sibbald and seasonal ground ice. I will also be looking into the CO2 fluxes at a peatland and event scale. Sibbald fen is exposed to Chinook events, which are transient air warming events that can lead to a total melt of the snow layer to the point of thawing the upper layer of the soil. We're interested in looking at these events and relating them with CO2 emissions measured by eddy covariance. And finally, I think it takes a village to do research, and this experiment definitely took a village. I want to thank all the people who were involved in this experiment and process in any sort of way. A big thank you to my supervisor, Sherry Westbrook, for her guidance. Sometimes I think she believes in my abilities more than I do myself. Thank you to the Coldwater Lab in Canmore and the Biogeosciences Institute of the University of Calgary for hosting us and serving as the hubs for our fieldwork. Finally, a huge thank you to my partner in life and adventures in science, Cobb, who hiked up a mountain with 20 kilo bags and boardwalks to measure these invisible things we call gases. And it takes a village, but it also takes money. So thank you to our funding agencies and projects. And thank you so much for your attention. I really hope that this presentation sparked your curiosity, and I will be ready to answer any questions to the best of my abilities.